Hi, everybody, and welcome to today's webinar. This is the first of two webinars celebrating the plant cell focus issue on climate change and plant abiotic stress, which came out in January 2023. Today's webinar is hosted by Nancy Eckardt and moderated by Mariana Artur, and we have three speakers who Mariana and Nan will introduce. This webinar will be recorded and posted to YouTube within a few days, and our YouTube channel has lots and lots of other recordings of science talks. You might like to see it. We'd like to thank the American Society of Plant Biologists and ASPB members for supporting this webinar series. And if you're interested in becoming a webinar, you can use the code PRESENTS10 to get 10% off your membership dues. Please put questions for the speakers into the questions box. If you have problems, you can email me, Mary Williams, at mwilliams@aspb.org. If you're having connection problems, just disconnect and try connecting again. And now I'm going to turn this over to Nan.
Okay, hi everyone. Thanks, Mary. So today's webinar is highlighting authors from the current plant cell focus issue on climate change and plant abiotic stress. And I'd like to just give a brief introduction to this focus issue. So we had in mind to run a focus issue on plant abiotic stress and decided to emphasize the relation to climate change for several reasons. First, of course, it's widely acknowledged that climate change will have devastating consequences for millions of people. And some of the most obvious effects are abiotic stresses, such as heat stress and drought, with serious consequences for many crops, as well as natural ecosystems and humans. And the latest Intergovernmental Panel on Climate Change report suggested that we need to limit global warming to 1.5 degrees above pre-industrial levels to avoid the most extreme effects. And we probably have less than 10 years in which to accomplish this goal. Or in fact, it may already be too late, and we need to focus on limiting to a less than a 2-degree increase. So time is running out on the climate change clock. And finally, a couple of years ago now, I heard Juenger at the Salk Institute give a talk on this issue, work that they're doing at the Salk Institute. And she encouraged the audience of plant scientists to spend at least 5 minutes per day thinking about how your research can contribute to mitigating the effects of climate change. So with that in mind, we thought that it was important to add our voice and the journal's voice to those calling for action, and perhaps in this small way to provide information and examples of plant research in this area to encourage others to think about how your research might contribute. So the issue includes a commentary from two of our editors, Tom Juenger and Paul Verslues, that includes discussion and guidelines for conducting plant drought stress experiments. And then we have two what we call vignette style reviews, one that I organized with over 24 co-authors, where we discuss examples of research aimed at engineering efficient carbon capturing and carbon sequestering plants and developing crops with enhanced climate resilience, and another authored by Paul Verslues and co-authors that discusses unresolved questions in plant abiotic stress biology posed by 15 different research groups. And then we have another 6 in-depth review articles, a couple focused on different aspects of gene by environment interactions, a couple more on genetic manipulation and breeding for improved climate resilience, and one on abiotic stress memory in plants, and one on the cell biology of primary cell walls during salt stress. And we'll hear more on this from first author Leia Colin today. And finally, we have 6 research articles, a gene discovery pipeline for complex quantitative traits in maize that uses multiplex CRISPR-Cas9 genome editing, and a couple of articles that deal with the regulation of stomatal closure, and a couple more related to salt stress, and one on a vacuolar nitrate transporter that affects nitrogen use efficiency. And we're going to hear from the authors of 2 of these papers today as well, those I highlighted in purple here. And with that, I'll turn it over to our moderator, Mariana Artur, to introduce our first speaker.
Thank you, Nan. Thank you, Mary. I'm very excited to be here today to moderate this nice webinar with 3 very interesting talks. So good morning, good afternoon, good evening, everyone watching. So welcome. If you have any questions, please remember there is a Q&A box below, so feel free to add your questions. But first, I would like to introduce our first speaker of today, which is Dr. Leia Colin. Leia is a cell biologist with strong interest in microtubule-associated proteins and their roles on plant development and biology overall. She did her PhD in Olivier's Hamant group in Lyon, in France, where she investigated how the cell cortex contributes to mechanotransduction during plant development. During her PhD, she studied the impact of mechanical forces on microtubule organization at the cellular scale. And now she's a postdoctoral researcher at the Copenhagen University in the group of Staffan Pearson, where she works on plasma membrane cytoskeleton anchoring mechanisms. And today, she'll be highlighting her recent plant cell paper entitled The Cell Biology of Primary Cell Walls During Salt Stress. So welcome, Leia. The floor is yours.
Thanks. You see my screen?
Yes, that's clear.
Okay, perfect. So thank you for the nice introduction. It's my pleasure to be there today to talk about our last review, entitled The Cell Biology of Primary Cell Wall During Salt Stress. So since it's a review, it's a condensate of recent studies. So today I will be able only to talk about a few of them. But let's start first with a brief introduction about salt stress. So salt stress can drastically impact plant growth and development because it causes at the same time ionic stress, oxidative stress, and osmotic stress. And these 3 stresses can impact both the cell wall and the turgor pressure that are 2 critical parameters for cell expansion and cell growth. So in our review, we focused on the impact of salt stress on the primary cell wall. So the primary cell wall encases most of plant cells, and for this reason, it's one of the first barriers protecting the cells against biotic and abiotic stress, including salt stress. So the plant cell wall can be viewed as a hybrid material composed of the load-bearing cellulose microfibril embedded in a hydrated matrix composed of pectin, hemicellulose, and soluble protein. And the composition of this cell wall is really important as it has to be a strong but a pliable material to allow the cell growth. And in this cell wall, many components are directly impacted by salt stress, including the pectin and the cellulose microfibril. So today I will talk about only cellulose microfibril, since the cellulose microfibril largely determine the cell expansion, and we will see several strategies developed by the plants to sustain growth during salt stress. But first, let's remind you how the cellulose is sensitized. So the cellulose microfibril are sensitized directly at the plasma membrane by the cellulose synthase complex that you can see here, that is composed itself of cellulose synthase protein. And this cellulose synthase complex is connected to the microtubule cytoskeleton by the cellulose synthase interacting 1 protein. And during the process of the synthesis, this complex moves through the plasma membrane following the microtubule as a track. And with a red signal, I have indicated where this process can be impacted by salt stress. And here we can clearly see how salt stress can directly impact this process. So here you have some imaging of a cell with a reporter of cellulose synthase protein. And you can see that 2 hours after a salt treatment, the Caesar protein are not anymore at the plasma membrane. They have been internalized. And at the same time, microtubules have been depolymerized. However, 28 hours after a salt stress, a salt treatment, the CISA protein are back to the plasma membrane and the microtubules are repolymerized. So that indicates that the plants have developed strategies to recover growth after salt stress. And one of these strategies involved the companion of cellulose synthase protein, the CC protein. Indeed, in the double mutant CC1, CC2, you can see that 28 hours after salt stress, the cellulose synthase protein are not yet back at the plasma membrane, and the microtubules are still depolymerized. And interestingly, a recent paper published in 2022 in Science Advances I've involved another type of protein in a similar with a similar phenotype as a double mutant CC1, CC2. These proteins are the tetratricopeptide pterodoxin-like protein, the TTL. In the double mutant TTL1, TTL3, you can see also that 28 hours after the salt stress, the CISA protein are not back to the plasma membrane. However, these two proteins are involved is really in two distinct mechanisms. So here you have a schematic representation of a cell before salt stress. You can see in this case that TTL3 protein is located in the cytosol, while the CC1, CC2 protein is associated with the cellulose synthase complex at the plasma membrane and interact with microtubule. However, after a salt stress, you can see here that the TTL3 protein has been recruited to the plasma membrane and now interact with the cellulose synthase complex. While the CC1, CC2 protein has been internalized together with the cellulose synthase complex into small compartments. So the regulation of how the location of TTL3 is triggered by salt stress is still unknown and require more investigation. However, the molecular mechanism behind the CC1, CC2 process or mechanism has been well described in a more recent paper published by the same group in 2019. So in this paper, the authors have characterized the N-terminal region of the CC1 protein. They have identified 4 individual microtubule binding sites on the N-terminal region. And with in vitro assay, they have shown that this N-terminal region can trigger microtubule bundling. Then they have tried to, or they have addressed actually, the role of this interaction with microtubule in vivo and during salt stress. So to do so, they have replaced 2 tyrosine by 2 alanine on the first binding site with the microtubule on the CC1, and preventing the interaction of microtubule with this binding site on the CC1 protein. And the first thing that they observe is a decorrelation between the truncated version of the CC1 and the microtubule signal. Then to assess the role of this interaction between CC1 and microtubule during salt stress, they have performed this quantification. So here you can see the quantification of the microtubule bundling during a salt stress. So the bar indicate the microtubule bundling, and here you have the time after salt stress. So in dark gray, you have the wild-type version of the CC1 protein. And what you can see and observe actually is the increase in the microtubule bundling after a salt stress, while for the truncated version of the CC1 protein, you don't have this increase in the microtubule bundling. And in conclusion to this paper, the authors have shown that the CC1 protein can promote microtubule reassembly after salt stress, and in turn promote the growth recovery after salt stress. So I have talked about these 2 mechanism for the growth recovery after salt stress, and that involve different kind of protein. These experiments were conducted in hypocotyl cells. Now I would like to draw your attention on another mechanism, which is more about the maintenance of the growth during the salt stress. And in this case, it occurs in the root cells, and it's called the allotropism response. So the allotropism response is the capacity of a root to grow away from a salt-concentrated medium. It has been described for the first time in 2013. And how can you observe this response? So you have here a square plate divided in two main parts. The bottom part is MS medium supplemented with salt, while the upper part is only MS medium. So the salt will diffuse through the plates and will create a gradient. And for the col zero, when it's in contact with this gradient of salt stress, the roots will change its growth orientation, and you will have this root skewing phenotype. However, until this recent paper, until now, the underlying cellular and molecular mechanism that were regulating this allotropic response were still not clear. But this recent paper published in Developmental Cells have revealed new insight about the regulation. So here, this paper starts with the fact that salt stress can induce abscisic acid. So the authors have screened different ABA-related mutants to screen whether they were impaired in their root allotropic response. And they have identified several of them that cannot trigger this, that cannot respond to the cell stimulation, sorry. And these mutants were different kind of mutants. Some of them were signaling mutant, others were involved in the biosynthesis of ABA. So now we can ask how, why this mutant cannot respond to salt stress. And to answer this question, we need to focus a bit more about this skewing phenotype that occur in the wild type. So here you have a root that grow on a divided plates, so that experience a salt stress, and that has been segmented by the morphographic software. And what you can observe, basically, it's a change in the anisotropic growth and a cell-file rotation that in turn triggers a root skewing. So the anisotropic growth is largely controlled by the deposition of the cellulose microfibril in the cell wall. If the cellulose microfibril are in the transverse orientation in the cell will expand straight. While if you change this orientation in the cell wall, and now the cellulose microfibril are in the oblique orientation, you will induce a cell-file rotation and a change in the anisotropic growth. And as I mentioned earlier, the cellulose microfibril deposition largely depend on microtubule orientation. So in the next part, the authors have screened for protein targeted by ABA related protein that could regulate microtubule behavior. And they identified this microtubule associated protein spiral 2-like. And here you can see that the mutants of this protein is not able to respond to salt stimulation, and the root grows straight. You have exactly the same movie here, where you don't see any cell-file rotation and the anisotropic growth is still straight. So they were interested about the microtubule behavior in these roots, in these mutants. And what is really interesting here is, like, before the salt stress, both the wild type and the mutant display transverse orientation for their microtubule, while after 2 hours, after the salt stress stimulation, you can see that for the wild type, you had a reorganization of the microtubule. They are now in the oblique orientation. So we can know that the cellulose is also in the oblique orientation and triggers a change in the anisotropic growth. While for the mutants, microtubule are mainly still in the transverse orientation. That explains why the root continue to grow straight. And so they finally proposed this model in which there are way more information than what I gave you today. But basically, the salt stress can trigger the abscisic acid that can activate the SNRK2 protein, which is a kinase that can phosphorylate the sp12 protein. The phosphorylation of this sp12 protein triggers the microtubule reorientation during a salt stress. And the microtubule reorientation is responsible of the change of the anisotropic cell expansion through the deposition of the cellulose microfibril. So I would like to thank you for your attention, and also if you are interested, I would like also, to thank the other authors of this review. And if you are interested for more detail and other studies that are more focusing on pectin, you can go and check this review. Thank you.
Thank you, Leia, for the nice talk. So everyone, if you have questions, please type it in the Q&A box and I will read them for you. So in the meantime, I have a quick question while I check here the Q&A box for you, Leia. So you highlight in your work that the cell wall integrity and sensing is important is a key aspect for the salt sensing. And also that the cell wall composition varies among species and also in different tissues. So I was also wondering, in a developmental, but also in a cell biology point of view, are there studies showing that depending on the tissue type or the cell type, a certain mechanism of salt stress sensing is more prominent? Or in other words, is salt sensing type, cell type specific or tissue type specific?
I think it's really interesting, this question. For what I know, I don't know. So there are several studies that show that cell wall integrity is important, and each time it's conducted in a specific tissue. But I don't know if the highlighted mechanism is prominent to this tissue or not. And I'm not sure that there were some studies on this specific question, but it might be the case, specifically between the roots and the aerial part, I think, that are not exposed in the same way to the salt stress. So it could be that they have different way to respond.
Okay, thank you. We have a question from Monica Garcia Gomez, and that she says, nice talk, thank you. About the halotropism, does it mean that if the salt gradient comes from the opposite direction, the microtubule organization, cell file rotation, and skewing will happen in the opposite direction?
That's a really nice question. So if she means by opposite direction, we change the way to divide the plates. So not the salt on the upper part, but more on the left or the right. Actually, it seems that this response is observed only in this condition, and the root is not as well capable to respond if the stress is in the other direction, actually. So it's really interesting that it seems to be linked to the way how the plate is divided. I hope that it answers the question.
Interesting. Yeah. One more question from Michelle Bianchi. So would you say that root halotropism and hydrotropism are based on different mechanisms at the cellular level?
That's a nice question too. I should have revised my knowledge about hydrotropism. I think that we can separate these two mechanisms. They are not exactly the hydrotropism stress is based on the osmotic stress, while the halotropism response is based on the salt stress, so the ionic stress. So it can be, I think it is two different mechanisms. But yeah, I'm not totally sure.
Okay, thank you very much. There are a lot of questions in the Q&A, so that would be nice. Thank you everyone for asking. Leia, I think you have access to the Q&A, so maybe you can answer some of those questions in the meantime, or later on contact. So, yeah, thank you very much for the nice talk. So to keep track of the time, I would like to move on to our second speaker of today, which is Sophie Filleur. So Sophie received a PhD from the University of Pierre and Marie Curie and worked as a postdoctoral researcher at the Lancaster University in the UK. She has a long-standing interest in nitrate transporters and nitrate signaling in Arabidopsis, and she was also one of the pioneers in the characterization of the molecular mechanism involved in nitrate uptake by plant roots. She's now an assistant professor of plant biology at the University of Paris-Cité and does her research in the Institute for Integrative Cell Biology at the University of Paris-Saclay campus. Her research is currently focusing on the function of plant cell vacuoles in adjusting anion homeostasis, nitrogen and water use efficiencies. And today she will be highlighting her plant cell paper entitled Proton Exchange by the Vacuolar Nitrate Transporter CLCA is required for plant growth and nitrogen use efficiency. So welcome, Sophie. The floor is yours.
Sorry.
Yeah.
Okay, is it okay?
Yes.
Like this, yeah, okay.
Yeah, so maybe you can start. Yes, it's okay. Perfect. Thank you.
Okay, thanks Mariana for the nice introduction, and hello everybody, and thanks for the invitation to give this webinar. Then today I will talk on behalf of Julie Audin, who did a PhD with me, and I will introduce her work as she cannot be here today. Then first of all, I want to start with basic things, then to show, in fact, nitrogen, it's one of the main essential elements necessary for plant growth. Then it has been used extensively as nitrogen fertilizer to increase crop yield. However, the use of this fertilizer has been in excess compared to plant demand, which lead to environmental pollution, such as lake eutrophication. Then research needs to be done and to improve nitrogen use efficiency, especially as well in the context of global warming, as we know, this global warming decrease anyway. Then nitrogen is absorbed by the plant as nitrate. Once inside the root, is translocated to the shoot, where it is either stored in the vacuole or assimilated to amino acid. At later stage, nitrogen as nitrate, amino acid or protein can be remobilized towards the seeds. Then our team is focusing its research mainly on nitrate storage, as this compartment is very important for the plant to cope with nitrate fluctuation in the environment. A few years ago, we characterized a nitrate transporter called CLCA, and we show with the KO mutant for this protein, then this genotype under accumulate nitrate, and as well this KO mutant is highly sensitive to dehydration, like you can see in this short video. And this is mainly due to the fact this KO mutant contains less water. Then to summarize, we show finally, since CLCA is localized on the vacuolar membrane, is a nitrate proton exchanger, which is the main transporter responsible for nitrate storage in the vacuole. We showed as well nitrate is in the vacuole is very important to adjust the turgor pressure, as is responsible for mainly for the entry of water in the cell. Then CLCA is part of a very conserved family, and what is very interesting in this family is that you can find both exchanger like CLCA and channel. And other group are shown then the difference of exchanger and channel is due mainly to a glutamate, which is at the position 203 in CLCA. And it has been shown this glutamate with its side chain block the pore of the exchanger. But when you add proton, this proton link to this side chain and move it, which open up the pore of the exchanger and allows the nitrate to go through. If you mutated this glutamate into alanine, you convert this exchanger into a channel as the side chain of the alanine is not protonable and is very short as well. Then this result has been demonstrated using heterologous system, and us we wanted to see what are the physiological consequences of converting an exchanger into a channel. For that, we introduced the mutated form of CLCA under the control of 35S promoter into a KO mutant. We obtain different transgenic lines and we compare their phenotype to wild type, KO mutant, and the complemented line where we introduce the wild type form of CLCA. First of all, the main things we wanted to see is as CLCA is very important for nitrate storage, we analyze first the nitrate accumulation in this line. Then we've done that on plant grown on hydroponic and check first in this system the KO mutant still under accumulate nitrate in both shoot and root. But what was interesting in our transgenic line expressing the CLCA channel, this under accumulation is even higher in this line compared to the KO mutant in the shoot. Then to carry on and to better understand this phenotype, we decided to analyze the kinetic of nitrate accumulation by changing the nitrate availability in the medium. Then for that, we grow the plant on nitrate, starve them for 10 days, and put the plant back on nitrate for 120 minutes. And we did analyze this nitrate content in both shoot and root. Then in the KO mutant, the nitrate accumulation rate is still lower compared to the wild type and complementing line, both in shoot and root. And in the transgenic line expressing CLCA E203A, this rate is even lower compared to the KO mutant in the shoot. Then what we could say from this result finally is that in this, when you convert the exchanger into a channel, you reduce first the entry of nitrate in the vacuole. And second, then the nitrate, which is in the vacuole due to the activity of other transporter, it's leaking outside the vacuole, which explains, in fact, that this transgenic plant even under accumulate nitrate even more than the KO mutant. Then, as we reduce nitrate storage in this line, we wanted to see the result on nitrate homeostasis, as the plant cell can control the cytosolic nitrate concentration between 1 and 5 millimolar. And one of the main components to adjust this homeostasis is nitrate assimilation. Then we measure it in a rosette plant, and for that we analyze amino acid content, and protein content, and show again in this transgenic line you have approximately 40% more amino acid and 25% more protein compared to the other line. And interestingly, amongst all these amino acids, 2 of them, asparagine and glutamide, are amino acids which are highly mobile through the plant. Then at later stage, we found the same result, and as well, we found the consequence of the conversion of an exchanger to a channel. The consequence is you increase nitrogen concentration in the seed, which finally increase the nitrogen use efficiency, which is the relative content of nitrogen in dry seed, in this plant. Then if I sum up this first part of the result, finally we were very happy because finally the conversion of an exchanger to a channel shows that it can increase nitrate assimilation to amino acid, and by different result I don't have time to present, we show that lead to higher translocation of nitrogen to the seed and consequently to higher nitrogen use efficiency. Usually, when you increase nitrogen use efficiency, the plant is happy and grows better. However, in our case, it was not the case, as when we grew the plant, we've noticed that first the KO mutant are smaller than the wild type, but our transgenic line with CFCA E203A grows even less than the KO mutant. Here you've got a quantification of shoot and root to show that. To explain that, we decided to measure the water content in a plant. As I told you in the introduction, CFCA as well is important for this content. And surprisingly, we found this water content, a good correspondence of the decrease of water content in this line with the decrease of biomass. And this low water content leads to a plant which are very sensitive to dehydration. We also analyze the size of the mesophyll size using flow cytometry and show that this reduction of water content lead to a reduction of plant size. Then in conclusion, what we can say finally from this work, then we're exchanging, converting, the exchanger to a channel, increase nitrogen use efficiency. However, as you decrease nitrate content inside the vacuole, you decrease the entry of water inside the vacuole, which probably leads to a smaller cells and a smaller plant. Then the plant finally are more sensitive to water use efficiency. Anyway, in the future, we want to see if we can use this result for translational biology to crop. First, we would like to see if the mutation is dominant, which will make it easy to introduce in crop. And we want to see if we can increase nitrogen use efficiency without decreasing water use efficiency by expressing the mutated form of CFCA under different organ-specific promoter. And to finish this talk, I would like to thank all the persons who participate in this work, all the people of our team at I2BC, a team led by Sébastien Thomine, Mick, who did all the analysis by flow cytometry, Alexis, who did all the analysis of electrophysiology, result I didn't have time to show you today, and Fabian Anne, who did all the analysis for nitrogen use efficiency. And finally, I would like to thank all our funding sources and thanks for your attention.
Thank you very much, Sophie, for the nice talk. We have a few questions. Due to the time, I will ask you one, which is, thank you for the nice talk. Do you find any natural variation in CLCA203 in wild relatives? For example, does the E203A mutation occur conferring channel function in other plants?
It's a good question. In fact, we just had a quick look, not a systematic look. And at the moment, we cannot find any variation. But it was a quick look. Then we have to look more deeper on this.
Okay. Maybe I will ask another one very quickly from Burko sorry. Did you look at the water content of the shoots in the transgenic plants for CLCA, which accumulate less nitrate?
In the KO mutant, you mean? Or,
yeah, it says in the transgenic plants. I think it is in the knockout mutant. If you look at the
yes, it contains less water, but the difference is very low compared to the wild type and the plant expressing the channel.
Okay. Yeah, thank you very much for the talk. There are still some questions in the Q&A box, so if you could answer that for the people that are still asking some questions there, that would be great. Thank you, Sophie. Yeah. So now we'll move on for our last speaker of today, which is Dr. Yong-Fei Wang. He works at the CAS Center for Excellence in Molecular Plant Sciences in Shanghai, in China. And he received a PhD in botany from China Agricultural University in 2002 and worked as a postdoctoral researcher at the University of California, San Diego from 2003 to 2009. He started his laboratory at the CAS in Shanghai in 2009, and his group works in the field of ion signaling and nutrients in plants. And today, he will be highlighting his recent paper at the Plant Cell, entitled Multiple Cyclic Nucleotide-Gated Channels Function as ABA-Activated Calcium Channels Required for ABA-Induced Stomatal Closure in Arabidopsis. Dr. Yong-Fei, the floor is yours.
Yeah, thank you. Now I start share my screen. Yeah, okay. Can you see my slide?
Yes, it's on the way, I think. Let's wait a little bit.
Can you see my slide?
Yeah, now you can put in the presentation mode. Yes, perfect. Thank you.
Okay, thank you. I'm glad to be here to give a talk. And yeah, today my talk is about the calcium channels and the calcium signaling in guard cells. As we all know, the ABA triggers the cytosolic calcium signaling in the guard cells, and then to trigger the stomatal closure. And if we simply remove the external calcium or add some calcium channel blocker to inhibit the calcium efflux into the guard cells, and the ABA activated the calcium signaling in guard cells, and the stomatal closure will be inhibited. So the calcium channels in the plasma membrane of the guard cells play important roles in the generation and the regulation of the calcium signaling in guard cells for AB-induced stomatal closure. So this is an example of AB-induced calcium oscillation in the guard cells. But the calcium channels responsible for the calcium efflux are unknown for many years. So the identification of this calcium channel become a key step for us to understand how ABA initiate and regulate the calcium signaling in guard cells for ABA-induced stomatal closure. So there are several families. They are permeable to calcium. They could be the candidate of these calcium channels. And we feel that CGC families, they are the most possible candidates. So our research mainly focused on this family. CGC family, they basically form the tetramers, homo or hetero tetramers, and each subunit has a short N-terminal or 6 transmembrane domain and a long C-terminals. At the early stage of our study about the function of CGC channels in guard cells, we have analyzed a double mutant CGC5 and 6, but we failed to observe any clear AB-related phenotype of stomatal movement regulation. So we think the strong redundancy between multiple CGT members could be the main cause of our failure. So to overcome this difficulty, so we decided to used the dominant negative strategy to study the function of CNGC in GAR cells. The dominant negative means that if the mutated or non-functioning subunits, they are accumulated in a cell, and a large amount of the protein accumulated in the cell, and the protein will be integrated into the complex, the protein complex, to inhibit the channel activity. This strategy has been successfully used in the study of inward potassium channels in the GAR cells. So to do so, we first generated 3 mutated versions of CNGC6, which is known being highly expressed in GAR cells. And then we transiently express the different version of CNG6 in the Higgs cells. We did calcium imaging, and a PET clamping experiment. Here we can see the 3 mutated version. They show much smaller calcium channel activity, and the co-expression of the mutated version and the wild-type version will suppress the calcium channel activity of the wild-type version. And the PET clamping data also show a clear inhibition of the wild-type CNGC6 by the mutated version, and in a dose-dependent manner. So it looked like this strategy worked well, and then we overexpress this different version of CNG6 in the Abiotic plants in the background, wild-type background. So we simply call these lines expressing the mutated version as the DNA lines, and the lines overexpressing a wild-type CNG6, we call it the OE lines. So this is a AB-induced stomatal closure assay. So we can see the 3 DNA lines. They are obviously insensitive to ABA, but the OE lines show a slight AB hypersensitive phenotype. And this is a time course of 10 micromolar ABA-induced stomatal closure, and this is light-induced stomatal opening. And once again, we can see the 3 DNA lines. They are insensitive to ABA and cannot be inhibited by the ABA. And then to further identify the main CNGC members that's involved in this process, we generated a number of transgenic lines expressing the GAS genes under the promoter of CNGC families. And here we can see several CNGC members they express in GAR cells, including 5, 6, 9, 8, 12, and 15. And then we also conducted a QRT-PCR experiment, and 4 of them, their highly expression was confirmed by this data. So in our further experiment, we mainly focus on these 4 CNGC members, namely 5, 6, 9, and 12. So we based on this information, we generated a triple mutant and a quadruple mutant, and we eventually observed a strong ABA insensitive phenotype of stomatal closure and in the quadruple mutant. And we further generated the complementary lines, we simply call it the COM line, in the background of the quadruple mutant. And we also generated the OE lines by overexpressing a wild-type CNGC in the wild-type background of Arabidopsis. So, and then we did a stomatal assay, and this one is ABA light-induced stomatal opening, and we can see the quadruple mutant is the only one, which is insensitive to ABA, and the stomatal still can open, and all other lines, their opening was inhibited. In the AB-induced stomatal closure, once again, and the quadruple mutant is the only line showing clear AB insensitive phenotype. The common line behave just like wild type, and the OE lines show some AB hypersensitive phenotype. And this is a different concentration of ABA. And we generated a number of transgenic lines, including 8 common lines and 8 OE lines. So we summarize all the data, I mean the 10 micromolar ABA induced stomatal closure AC data in this figure. So here we can see this without ABA, the right side is with ABA. We can see that the quadruple mutant is the only one showing a strong ABA insensitive phenotype of stomatal closure. And then to confirm this phenotype, we use a different technique to analyze the phenotype. This is a fresh weight loss of with fresh weight loss analysis of detached leaves. And here we can see with or without ABA treatment, the quadruple mutant leaves, they are wilted faster than other lines. We can see here, here, and this one, and this one. The common lines and OE lines, they basically behave like, just like their response, just like wild type. And we also analyzed the drought stress tolerance in whole seedlings. And after 15 days drought stress and 7 days after being rewatered, we can see the wild type, the common line, and the OE line. Most of the plants can survive, but most of the plants of a quadruple mutant failed to survive and they died. And then next, we use the gas exchange technique to monitor the transpirational conductance change in the detached leaves. And the ABA can added in the solution in the small tubes absorbed by the leaves to induce stomatal closure. We divided our 18 lines in 4 groups, and the wild type and the quadruple mutant were included in each of the group. As we can see, the quadruple mutant show a clear AB insensitive phenotype, and stomatal close much slower than other lines. And the common lines, their response just like wild type, and the OE lines more or less, you see, more or less show some AB hypersensitive phenotype. So after we confirm the phenotype of AB-induced stomata closure and AB inhibition of light-induced stomata opening, and we conducted the patch clamping experiment in garden cell protoplasts using the barium-based Besson pipette solution. We can see in the wild type, common lines and OE lines, we can observe a clear ABA activation of inward calcium channel activity. But in the quadruple mutant and DN lines, the calcium channel current were largely inhibited. And finally, we directly monitor the calcium signaling in garden cells. We can see in most of the garden cell, a wild-type garden cell, we can observe calcium AB-induced calcium oscillation. But in the quadruple mutant, this normal calcium oscillation patterns was disrupted. And the common lines, all the common lines, their response is just like wild type. And also the OE lines also they show a wild type like calcium oscillation. So we take out we also statistically analyzed this data, and this figure we can see the average duration of calcium peaks is almost doubled. It's much longer than the wild type and other lines, including COM and the OE lines. But the average magnitude of calcium increases was reduced by about 50%. So that means the normal calcium oscillation pattern was disrupted in this mutant. So take all the data together, we believe the multiple CGC members, they redundantly function as AB activity, the calcium channels, which is required for AB-induced calcium signaling and then further induced stomata closure. So at the end of this talk, I would like to thank my team members, the three names in the red color. They did the experiment. I would also like to thank my collaborators for their help and the technical support and also the funding. Thank you for your time, and let me know if you have any questions.
Thank you very much for the very nice talk. We have a few questions in the chat box, so I'll read the first one. Is the same mechanism of CNGC in other plants like Rhabdopsis, or I think the question is more, is this protein in other species performing similar functions in other species?
So my answer, I think yes. I think yes. Yeah, there's no so much papers in report their function and in other species, but I think the answer should be yes.
Okay, thank you. And we have a question from Mark Knight. Some of the overexpression lines show hypersensitivity to ABA with respect to guard cell closure, but the calcium signatures are the same. So how does this increased sensitivity work?
I'm sorry, can you repeat it? Repeat the question?
Yeah, so he's asking how the increased sensitivity of the plants happen, because it seems that some of the overexpression lines show hypersensitivity to ABA.
Yeah.
But the calcium signatures are the same.
Yeah, I guess that is because the accuracy of the data. Some experiment they may not sensitive enough to response to show the difference, and the calcium oscillation, this data they may not be accurate enough, and they cannot show the slight difference, the slight hypersensitive. I think that is the reason. Yes.
Okay, interesting. There's one more question. I think we have time for one more. Bello Hassan is asking if is there any interacting partners of CNGC?
We didn't test that. We don't know yet.
Okay. Any hypothesis? I saw in the research paper that the future work that you're planning to investigate is the molecular function of this protein. So do you think that they could interact with other proteins, or not really?
Yeah, we have projects going on in my lab. Yeah, we are working on it.
Okay, interesting. Yeah, thank you very much. I think we are on time. I believe we kept the time schedule well, so I would like to give the word back to Mary, and thank you for the very nice talks, all these speakers. Thank you.
Absolutely. Thank you for 3 excellent talks, excellent hosting, excellent moderation. We have another webinar. We'll be advertising it starting tomorrow on February 22nd with 3 more papers from this excellent focus issue. We hope to see you there. As I mentioned, we'll post this recording in a few days. And thank you everyone for attending and joining us, and have a great rest of your day. Thanks again. Bye-bye.
Bye-bye.